Pharmacodynamics refers to the mechanisms and effects of medications within the body. Or more simply, it's what medications do to the body and how they do it. Alright, so in order to have an effect, many medications have to reach their target cells and bind to a receptor. Receptors are specialized proteins found inside the cell or on its membrane. When they bind to a signal molecule called a ligand, they can alter their shape or activity, which ultimately results in some change in the cell's activity or behavior. You can imagine the ligand as the key that fits into the lock, which is the receptor, causing it to open or activate. Now, depending on the effect a medication has on its receptor, they are often divided into two major categories: agonists and antagonists. An agonist is a medication that mimics the action of the signal ligand by binding to and activating a receptor. On the other hand, an antagonist is a medication that typically binds to a receptor without activating them, but instead decreases the receptor's ability to be activated by other agonists. Okay, now the maximal effect or response an agonist can produce, abbreviated as Emax, is determined both by the number of receptors bound to the agonist, which depends mainly on the amount of the agonist given, also known as dose, as well as its intrinsic activity, which is the ability of the agonist to fully or partially activate its receptors. So let's plot all of this into a nice graph to show the relationship between the dose given on the x-axis, usually on a logarithmic scale, and the response produced on the y-axis. So full agonists, upon binding to the receptor at high doses, are capable of producing a maximal response of 100% Emax on the y-axis. This represents the point where all available receptors are bound to an agonist. In contrast, partial agonists, even at a very high dose, result in a smaller response, so their Emax will be lower. For example, a 70% response would shift the curve downwards. In other words, a full agonist is like a really well-made spare key that's just as effective as the original, while a partial agonist is a poorly made spare key that could open the lock, but it takes longer and it gets stuck and you have to jiggle it around a little bit. Alright, at the other end of the spectrum, antagonists can be divided into competitive antagonists and non-competitive antagonists. A competitive antagonist will bind to the exact location where the ligand is supposed to bind, kind of like putting the wrong key into the keyhole, which prevents the right key from being used. Now, competitive antagonists can be reversible, where they can unbind from the receptor after a while, or irreversible, where they bind to the receptor permanently. The inhibition caused by reversible competitive antagonists can be overcome when there are more ligands floating around. So, when it unbinds, it's more likely for one of the ligands to bind. So, on the graph, reversible competitive antagonists typically shift the curve to the right without affecting the Emax, but they increase the dose required to achieve 50% of the maximum effect, also known as the effective dose, ED50. In other words, this type of antagonist influences the dose of agonist needed to elicit a maximal response, but doesn't affect the extent of that maximal response. Now, an irreversible competitive antagonist also competes with the agonist for the same receptor site, but the binding is irreversible. This means that the receptor will not activate, no matter how many agonists are around. What we see on the graph is a right and downward shift of the curve of the agonist with both an increased effective dose and a reduced maximal effect. On the other hand, non-competitive antagonists don't bind to the same site as an agonist. They bind to a spot called an allosteric site. And when they bind, it causes the shape of the receptor to alter in such a way that the ligand can no longer recognize it as a binding site. So, imagine a giant spike being driven into the lock, causing it to crack and rendering it useless. And just like in irreversible competitive antagonists, Even if we increase the levels of the ligand, this inhibition cannot be overcome. So, once again, the curve shifts to the right, increasing the effective dose, and down, reducing the maximal effect. In other words, both irreversible competitive and non competitive antagonists reduce the extent of the maximum response that can be achieved by any dose of the agonist. Alright, let's switch it up again and talk about other not so classical forms of antagonism, which include physiological or functional antagonism, where the antagonist acts at a completely different receptor, producing a response that is physiologically or functionally opposing the agonist, meaning that they cancel each other out. A typical example would be epinephrine, which acts on beta 2 adrenoreceptors on bronchial smooth muscle, causing them to relax, and antagonizes histamine, which binds to H1 histamine receptors, causing bronchoconstriction. Likewise, there's chemical antagonism, where the antagonist forms a chemical complex with the agonist, reducing the agonist concentration. Finally, there's pharmacokinetic antagonism, where the antagonist influences the absorption, distribution, metabolism, or excretion of the agonist. Alright, as a quick recap. Agonists activate receptors and can be divided into full agonists, which can produce a maximal response, and partial agonists, which can only produce a submaximal response. Antagonists prevent receptor activation and can be reversible and competitive, meaning they bind reversibly to the same agonist binding site, irreversible and competitive, meaning they bind irreversibly to the same agonist binding site, and non-competitive or allosteric, meaning they bind at a different site but still prevent the action of the agonist. Reversible competitive antagonism can be overcome by adding more agonist, while both irreversible competitive and non-competitive antagonism cannot. Lastly, there are physiological or functional antagonists, chemical antagonists, and pharmacokinetic antagonists as well.